mycelium of mucor. Yes, you
Yes, sir.
understand
Yes, sir.
Yes,
the
sir.
structure.
Yes, sir.
So in exam also you write first of all classification. The classification is division Eumycota, subdivision is Zygomycotina, class is Zygomycetes, order is Mucorales, family is Mucoraceae, genus is Mucor. So coming to structure, the mucor is a saprophytic fungus commonly found in the commonly found in the
Organ system.
commonly found in the soil and
Soil and the dung, organic materials.
Material.
dung also, and
Yes,
also
organic
organic
materials.
matter.
Humus.
The mycelium, where it is, the mycelium is forms below side is entire part is the mycelium. It forms loosely puffy masks of cotton over the infected substratum. The hyphae are prostrate. Some of the hyphae penetrate to the substratum. Penetrate means
Getting into, sir.
Attached to another,
attached to the
Injecting to the body.
one to
Other
another part.
poster.
Yes.
life cycle. Just what I share the life cycle. So here the mycelium is accepted and phenotypic. The septa are found when reproductive structures are just wait. I will trying to life cycle of mucor. So this is the mucor. You find out the diagram. Yes.
Yes, sir.
So this is the mycelium of mucor. The mycelium is septate and cenocytic. Septa are found when reproductive structure are developed. What are the reproductive structures here?
Sporangium.
Sporangium. The sporangium release the sporangiophores.
Sporangiophores.
Okay.
Yes, sir.
Spores, not four. The
Sports.
sporangium release
Sporangiophores.
the sporangiospores.
Sporangiophores.
Okay. These are the prostrate hyphae, subpenetrate hyphae. It is attached to the another substratum. So it is like aerial hyphae. It is the sporangiophore. Sporangiophore and sporangiospore is different. These are the some upright structure some hyphae grow in the upright and they are called the sporangiophore. Okay. This one, it is Calmanella. So next, these are the taxonomic fungi: division, mycota, zygomycota, mucorales, and muceraceae, mucor. Habitat. So it is appears radial in the dump shops, dung, when already we discussed, and it is found in the soil, dung under the organic materials. The dung, when kept under the bell or jar, it is immediately appears like bread. You know, just you keep in a side after expiry date of this bread, 1 or 2 days, leave it in 1 or 2 days. This type of formation is appears. What is this? This is the mucor. When
Mucor.
this, you know, already everyone see this mucor on the breads. Bread,
Sir,
especially.
Bread mold.
Rhizopus.
Rhizopus, sir.
So
Bread is
so
in
same
rhizopus.
rhizopus, mucor is the same na, like brother and sisters or sisters.
Okay.
Rhizopus, mucor under only one category. The difference between, what is the difference between some sexual reproduction? These are different. The remaining things are the same. Okay, clear?
Yes, sir.
Rhizopus
Okay,
and mucor, it is like brothers or sisters.
Brothers and sisters.
Okay, it is simply just not only so some of the exams they are asking the mucor, it is also
Rhizopus. Bread mold.
here. We will see under the microscope. These are the structures. Find out. These are the structures. Yet it is able to see this one.
Yes, sir. Yes, sir.
So next, coming to vegetative structure. The mycelium consists of numerous slender, freely branched filamentous called hyphae. What is meaning of hyphae? Already we discussed in the general character of fungi. All cover these points like hyphae, penetrate, okay, substrate. Okay, so the mycelium consists of numerous slender, freely branched filaments. Freely filaments called hyphae. The hyphae form a fluffy mass, white in color, called as mycelium. After mycelium formation, the reproductive phase starts. Like black pin-like structures. Black pin-like structures appear among mycelium, called is sporangia. So black-like. Yes, after mycelium formation, reproductive phase starts. Black pin-like structures appears on the mycelium, called that is the sporangia. The hyphae are phenotypic and accepted. The vegetative stage of the hyphae consists scrollon horizontal hyphae, scrollon horizontal hyphae, and rhizidal hyphae. So the rhizidal hyphae helps in the absorption of the nutrients from the substrate. The cell wall is made up of the chitin and cellulose. So here, just wait. Okay. Here the reserve food material is the What is this reserve food material? Is the glycogen and oil droplets. The cell wall is made up of the chitin and cellulose is absent. The organelles like mitochondria, endoplasmic reticulum are present in the cells. So next we'll go for these are the some vegetative mycelium, paraffin of hyphae under the light microscope, portion of the hyphae under electron microscope. Who talking? Just wait, I'll move that person.
Vijay Lakshmi, please mute.
Vijay Lakshmi.
Okay.
Vijay Lakshmi garu. Okay. So these are the diagrams of the vegetative mycelium and the portion of the hyphae under the light microscope. In the B, the C, it is a portion of hyphae under electron microscope. So A is vegetative mycelium. These are the A. And the left side one is the B, portion of hyphae under under light microscope. In the C, it is the electron microscope. So what are the components? Is nucleus present, plasma membrane, mitochondria, endoplasmic reticulum, ribosomes, vacuole, cell wall.
Cell wall.
These are the components present in this mucous cell. Next, same sporangium, columella. It is like pin-like structures. So how it is spores are released? These are the sporangium. The sporangium when breaks down, it is released the sporangiospores. Okay.
Yes, sir.
Vegetative reproduction. Coming to the vegetative reproduction. What are the vegetative reproduction in the common algae members? Sorry, common fungi members. Vegetative fragmentation.
So nothing like a fragmentation, budding.
Yes, fragmentation. So these are the some
Fragmentation, fission, budding.
Fragmentation. So when the separation of hyphae occurs from the aerial portion, then these fragments are capable of producing new hyphae, and this is called fragmentation. Very simple. The fragmentation, it is separation of hyphae occurs from the aerial portion. Aerial means? Yes. Aerial. Yes. Anyone? Aerial. My voice is audible?
Yes, sir.
My audio is audible. My voice is audible. Yes or no?
Yes, sir.
Yes, sir.
So what is the aerial parts?
Tentacles.
Yes. What is the aerial parts?
Hyphae.
Upper part.
It is like leaves, stems, all
That
these
is
things.
Aerial. What are the exactly fragmentation? The aerial portions that like small leaves, buds and stems, whatever may be, are capable producing. Well, then these fragments are, for example, the area separation of the hyphae from the aerial portions. Then the fragments are capable producing the new hyphae, and this is called fragmentation. Right? Clear? Next, chlamydospore formation. Chlamydospore formation under unfavorable condition. What is the unfavorable condition means? Yes, unfavorable conditions.
Critical.
Yes.
An adequate amount of food materials are not
suitable
available.
conditions.
Yes.
Unsuitable conditions, sir.
Yes. So favorable condition, it is opposite conditions of the unfavorable. It is favorable conditions.
Yes, sir.
Yes. So when
Sir.
the chlamydospore formation, when the conditions are under the unfavorable, the fungus produce the special modified thick walled resting cells called as the chlamydospores. Chlamydospores. It is under the vegetative reproduction. In this vegetative reproduction, the modified thick walled and resting cells, it is called as the chlamydospores. When will it is produce the chlamydospores? There is no favorable condition or unfavorable condition. That time the fungus produce special modified thick walled and resting cells are called as chlamydospores. They contain food and are very resistant to the desiccation. What is the meaning of desiccation? Yes, desiccation. Yes. Anyone? Desiccation. Desiccation. Yes, anyone?
I don't know, sir.
Yes.
Dries.
Yes, your voice is not clearly. Amma.
Drying, sir.
Yes. What about others?
I don't know, sir.
Try. It is very
Dryness.
simple terminology. Desiccation.
Dryness.
Yes.
Change into dry.
So your voice also.
Removal, moisturing.
Yes. Yes.
Removal, moisturing.
From the
Food.
Food.
So sorry, so many problems today. So I don't know why it is meeting suddenly off. So uh, asexual reproduction. So what are the reproduction organs in the mucor? Just tell me.
Sporangium.
Sporangium, spores.
Spores.
Right.
Sporangiospores.
Simply you recognize asexual reproduction in mucor. Only two things are there: sporangia, sporangia, sporangiophore, and sporangia and sporangiospores. Right? These three things. First of all, the mycelium, the hyphae, it is growing up the upright, like a straight line. That is called as sporangiophore. The sporangiophore, it is consist sporangium. Sporangium produce the sporangiospores. These are the very important in the asexual reproduction of the uh, reproductive organs. So it is asexual reproduction is placed by the formation of non-motile multinucleate. These sporangiophores are the multinucleate. The sporangiospore, multinucleate sporangiospore. The sporangia arise a single end terminally. Where it is located terminally means the pinna. The terminally at the tip of the sporangiospore. So the young sporangium is white and spherical in structure. The maturity at maturity time, it turns into the black color due to the formation of the spores. When the spores are formed, this color is black color. Okay. In the center of spore, there is a large dome-shaped structure called columella. This one.
Yes,
This
sir.
one. In the center of spore, there is a large dome-shaped structure called is columella.
Columella.
The sporangia are haploid. The sporangia are haploid.
Haploid.
So what is the meaning of haploid? It is the one set of chromosome. They are produced in haploid mycelium. The produced mycelium, it is also haploid condition and produce haploid spores. So here entire the asexual reproduction, this plant is haploid, right? The sporangia haploid, the produced haploid also haploid mycelium, the spores also haploid condition. Okay, here mucor in the 4 or 5 important points. First one is the sporangiosphore, next sporangium, sporangiospores, and columella. The columella and the sporangium color is the white and spherical in shape. The sporangium, it is in the maturity stage.
Black color.
Black color due to the formation of the spores. These are the regarding about asexual reproduction. So then we will go for the spores. How the spores after releasing of the sporangia, when break down, then release the spores, sporangiospores. How it is structured, how it is dispersed. The spores are dispersed after the rupturing of the sporangia. They are dispersed by the wind. Okay, clear? The spores are dispersed after the rupturing of the sporangia. They are displaced by the wind. The spore is non-motile. Non-motile means not movable, and it is oval shape, and it contains protoplast surrounded by the single layered cell wall. These are the regarding about the spore structure. Spore structure. So, and under suitable conditions, the spore germinates into form a germ tube. The hyphae arise from the germination tube and spread in all the directions. Clear? These are the regarding about the spores.
Spores.
Okay.
Yes, sir.
Next, sexual reproduction. The sexual reproduction, it takes place by the gametangial copulation. So what is the gametangial copulation? Already we discussed in the general characters of fungi or classification of fungi. We will, what is the gametangial copulation? Anyone?
Fusion of gametes.
I think we discussed it in the previous class, right? It takes place by the gametangial copulation. Just you recall to the what is the gametangial copulation, what is the gametangial collection, you find out. So the fusion gametes are the multinucleate and are distinct from the each other. These are the one important character. The fusion gametes are the multinucleate and are distinct from the each other. The sexual process starts within the formation of two special hyphae called zygospores. The sexual process starts with the formation of two special hyphae called zygospores. They arise from near the apices of the actively growing somatic hyphae of positive and negative strains. They are chemically induced. The zygospores meet and adhere in pairs at the tip to form the fusion septum. The tip swells due to the flow of nuclei and cytoplasm in them. It is called the progametangia. The progametangia combine to form prozygosporangium. Prozygosporangium. It's containing many diploid nuclei called zygospore. In the sexual reproduction, what are the main points? Is the formation of zygospores. Yes?
Yes, sir.
The hyphae, two different hyphae, it is progametangia. It connect to each other. So it is formation of progametangia. It is then formation of gametangia. The gametangia, it is finally formation of as a zygospores. Right? Clear?
Zygospores.
Zygospores. These are the
Yes, sir.
sexual reproduction of the mucor. Next, so here, this is the life cycle of sexual life cycle of mucor. What are the spores or formation? After chlamydospores, then the germination completed. Then they are go for the progametangia. Progametangia, then it is actually what is the gametangia? Two cells contact each other. Then in the finally the formation of only one cell, it is called as zygospore. Right? That is the gametangial. So here, this is suspensor. It is the conjugating the isogametangia. Then the formation of zygospore. Here, the zygospore, it is consists of the left and right side is the suspensors are there. Suspensors. This left side and the right side, what we call suspensor. Okay. Then this zygospore germinating and the here the germinating zygospores consist of the germ sporangiophore and germ sporangium. Then it is meiospores, meiospore germination. The finally it is the formation of the new mycelium. These are the sexual cycle of the mucor. Right? Very simple. What are the just the outlook of this one? these are the some habitat, taxonomic position, and so this is habitat. This is apparently everyone knows this one. So the vegetative structure also important. So what are the vegetative structure of mucor? The mycelium consists of numerous, just once again I repeat, because it is very, very important. So it is in theory and practical also. So when you take the fungus material, you observe under the microscope, these characters of the find out in the as a vegetative structure. Then some of the zygospores, they are permanent slides there. Just keep a slide under the microscope, just you observe, then you identify the, oh, this is the zygospore. These zygospores, it belongs to the mucor family, right? So this is the sexual reproduction. Regarding the sexual reproduction, the formation of zygospores in the sexual reproductions, okay? And the asexual reproduction, what happening in the asexual reproduction, the reproductive structures is the sporangiophore to sporangia.
Sporangia.
The sporangia consists spores. The spores, it is called as sporangiospores, and one dome-shaped part is there in the central. It is called as
Columella.
columella. These are the asexual reproduction. So once I repeat this one, vegetative. So mycelium consists of numerous slender, fleshy, branched filamentous. Branched means you just observe the diagram. Branched. How many branches? 1, 2, 3, 4, 5 branches are there. So the hyphae form a fluffy mass, white in color, called as mycelium. So after mycelium formation, reproductive phase starts. The structure of mucor, the black pin structures, black pins like gundu pinnu etluntaiyamma, gundu pinnudu. Black pins like structures appear on the mycelium called sporangia. So this, some of the hyphae, it is growing or developed. It is a upstraight or upright condition, or straight. It is growing in the straight. That what we call is sporangiophore. So the sporangiophore, it is consist of the sporangium. The hyphae are phenotypic and aseptate. At the vegetative stage, the hyphae is consist of stolen horizontal hyphae and the rhizoidal hyphae. The rhizoidal hyphae helps in the absorption of the nutrients from the substrate. The cell wall is made up of chitin and cellulose is absent. Cellulose is the absent, and the cell wall is made up of the chitin. The organelles, cell organelles like mitochondria, endoplasmic reticulum, these are the present of the cell wall. Next, these are the diagrams. Vegetative mycelium, and under microscope, under light microscope, these are the structure, and C is the portion of the hyphae under the electron microscope. So here the nucleus, plasma membrane, mitochondria, endoplasmic reticulum, ribosomes, vacuoles, cell wall. These are the regarding about vegetative structure. Next, these are the some wall of sporangia, sporangiophores, columella, apophysis, septum, sporangiophore, substrate mycelium. So it is vegetative reproduction. The vegetative reproduction, it is the fragmentation. Okay. When the separation of the hyphae occurs from the area, fragments are the capable of the producing the new plant hyphae and is called as the fragmentation. In this mucor, the vegetative reproduction in the two ways. One is the fragmentation, another one is the chlamydospore formation. Chlamydospore formation. unfavorable condition, the fungus produce special modified thick-walled resting cells called as chlamydospores, right? So they contain food and are very resistant to the desiccation. When conditions become favorable, it germinates to form a new mycelium. These are the diagram of vegetative reproduction through the fragmentation, through the chlamydospore formation. Next, asexual reproduction. Sporangia, already I repeated 2-3 times. Here, it is takes place by the formation of non-motile multinucleate sporangiophore. These sporangia arise singly and terminally at the tip of sporangiospore. The young sporangium is white spherical structure. The maturity it turns into the black color
Black color.
due to the formation of the spores. So, the sporangia are applied, they are produced in the applied mycelium, are produced in the applied
Spores.
spores also. So these are the structure of spores. Spores are dispersed after the rupturing of the sporangia. They are dispersed by wind. The spore is non-motile, oval, and it contains protoplast surrounded by the single-layered cell wall. Single-layered cell wall. Under suitable condition, the spore germinates into form a germ tube. The hyphae arise from the germination tube and spread in all the directions. So that is all the spore structure. Next, sexual reproduction. It takes place by the gametangial copulation. Very, very important. The sexual reproduction of mucor is gametangial copulation. The fusion of gametes or multinucleate are distinct from each other. The sexual process starts with the formation of two special hyphae called zygospores. They arise from near the apices of the actively growing somatic hyphae of positive and negative strands. They are chemically induced. The zygospores meet and adhere in pairs at the tip to form of the fusion septum. So here the tip swells due to the flow and nucleant cytoplasm. It is called as the progametangia. The progametangia combine to form prozygosporangium containing many diploid nucleal called as zygospore. These are the diagram of sexual reproduction of the mucor. So these are the today class. Tomorrow, day after tomorrow, we will go for the any one of you just list out the what are the examples are there. Class and type studies according to your book, textbook. Just tomorrow you send me tomorrow. It is easy for the next class. Actually, I don't have any books. Like actually in the last year also I teach some of the copies are there. That's why I able to reach you. Like this number is 210. That's why. So any of you just write here in on paper what are the type studies are there. Just list out. According to that, we will go for the studies. Okay. So next class, I think just you search. So in this, we will discuss in the Saccharomyces. It is the subdivision is the class is the Ascomycotina or Ascomycetes. Okay. In this example is the genus is the Saccharomyces. In this, we'll go for the in this very, very important. And next class is the Penicillium. Okay.
Yes.
So any type studies, these are the you follow in exam or practical exam. Come with first of all classification, compulsory taxonomic position or taxonomic classification of either fungi, either algae, either any plant. Compulsory classification is must and should. Then you go for habitat. Next, vegetative structure. Next, vegetative reproduction. After vegetative structure, directly they are go for the vegetative reproduction. Just types fragmentation and chlamydo spore formation. And next one is the asexual reproduction. And next one is the structure of spores, how the structure it is in the spores. So, and next one is the sexual reproduction. And finally, you draw the sexual cycle or life cycle of the mucor. Okay. The sex cycle, it is always, it is like sexual reproduction. Okay, don't confuse. The life cycle also, you start with the life cycle, entire life cycle, you start with the vegetative reproduction, then asexual reproduction, then sexual reproduction. These, when complete the sexual reproduction, the entire sexual reproduction is completed. Okay, these are the life cycle of the mucor. So here in the next class we will discuss in the penicillium also, and another one is Phaeagia, it is Ascomycetina. So just you, any one of you, just take the what are the type studies list on your book, just note down, please WhatsApp me. So we will go for the according to that. It is easy. So in case in the offline class, we don't have directly, we came to class, we take in the book, then we'll go for, but in the online classes, we have no access to the department. But so that's why anyone of you, I'm requesting you, please send me the type studies, just names like mucor, like penicillium, number 1 mucor, number 2 rhizophus, number 3 penicillium. So this type of how many type studies are there? Just listed. Please send to me. I will in the next class onwards we will go for that list only. Okay. So in this type studies, one is Deuteromycotina is Cercospora. Cercospora. You know Cercospora?
Listen, but not.
Yes.
Cercospora rachidicola, some name
Yes.
is there.
Generally, the Cercospora, 70 species in the genus of Cercospora, which cause leaf spot diseases. And various hosts belonging to the some cereals, pulses, oil seeds, fiber crops, vegetables.
Graminate family.
Yes.
Yes. Wheat.
So it is also very, very important. And your practical, it is in the major experiment. So next one is in this Politotrichum. This is also one of the major practical. And next one is the one important topic is the mushroom cultivation. How mushroom cultivation? It is there on your textbooks. Just you find out the mushroom techniques are available in your textbook. So in case it is available, I will go through that. So otherwise, I will prepare a separate one PPT mushroom cultivations. So regarding these the type studies, the remaining in the some Candida, albugo candida also is very important. This is also some Eumycotina, like Eumycetes. Under the Eumycetes, it is albugo and Rhizopus and also Mucor. These three are the under Mastigomycotina, Eumycetes members, and Ascomycotina. What are the example? Saccharomyces. Type studies. Just you find out and cross check with this list also. Saccharomyces, Penicillium. These lists are available in your book or not. Just anyone please send me the list. So in case not available in your syllabus, then we will also remove it. Okay. So next, so these are the type studies of fungi. So remaining 5 classes are there, more 5 classes. Yes. Yes, anyone please respond. Are you there?
Yes, sir.
Sir.
Yes, anyone, please send me the, my voice is audible. Please anyone send me the list, will completed according to our slots. Like our last class is the 27th. Na? So remaining classes, the 5 classes are there. Yes or no?
Okay, sir.
Yes or no? How many classes remain?
4, sir.
4, only 4?
Yes, sir.
Okay, the last date is the 27th, right?
For C1, it is 21, sir.
C1 is 21 only. Okay.
Yes, sir.
Okay. Right. So, any doubts regarding this class? Yes. Any doubts?
Yes.
Okay.
No, sir.
Okay. Thank you.
Thank you, sir.
Thank you, sir.